Hello everyone. A very good morning to all the participants, faculty members and students. On behalf of Dr. Subhash Technical Campus, I, Grishma Dalal, Assistant Professor, Faculty of Pharmacy and moderator of today's event, welcome you all in today's webinar on Mucosal Drug Delivery System by Dr. Jalpa S. Pon. There are few instructions for all the participants. For any query or doubts related to the topic, please write your questions in the comment box and I will convey your questions to the expert. The link for the feedback will be given at the end of the webinar and the certificate will be mailed to you after the submission of feedback form. So now let me introduce, it gives me an immense pleasure to introduce and welcome Dr. Jalpa S. Pon. Dr. Jalpa did her PhD in pharmacy from Gujarat Technological University, Masters in Pharmaceutic subject from Department of Pharmaceutical Science, Saurashtra University, Rajkot, Masters in Ayurved from Gujarat Ayurved University, Jamnagar, and Bachelor's from B.K. Modi Government Pharmacy College, Rajkot. She has experience of 13 years. She has supervised 7 master's students and also completed minor research project funded by good cause. She has also filed patent and also is a member of life member of Association Pharmaceutical Teachers of India, that is APTE, and Indian Society of Technical Education. With this brief introduction, I would like to invite Dr. Jalpa Pon to take the charge of this webinar on mucosal drug delivery system. Please, over to you, ma'am, sir.
Okay. Good morning, all of you. I am very much thankful to Dr. Subha's Technical Campus and its organizing committee for giving me such an opportunity to share my views on mucosal drug delivery system, and I am lucky enough to have such great audience with me. Let us start with my presentation on mucosal drug delivery system. Slide sharing is confirmed with you, ma'am? Hello? Hello?
Yes, ma'am, it is showing.
Okay, good.
Okay.
So let us start with content of my topic, that is introduction principle of bioadhesion, which are the steps of mucoadhesion that are involved, some advantages and disadvantages of mucosal drug delivery system, transmucosal permeability, formulation consideration of buccal delivery system, some evaluation parameter of the doses form, and finally the conclusion. Let us start with a brief introduction about the doses form. A bioadhesion word can be defined as the state in which two materials, at least one of which should be a biological in nature, and that's why term biological is utilized for the bioadhesion word, and it is held together for extended period of time by the interfacial forces. In the pharmaceutical sciences, when adhesive attachment is performed to the mucus or a mucous membrane, the phenomena is known as mucoadhesion. So, bioadhesion and mucoadhesions are generally considered as a synonym for pharmaceutical field. So whenever comes a mucoadhesion term, it is coming from mucus, and the content of mucus is having 95 to 99% of water, which contains 0.5 to 5% of mucin and some other substance like immunoglobulin A, lysosome, lactoferrin, some salts, growth factors, trefoil peptides are included. From this mucin, specifically from which a word is obtained, so it is heavily glycosylated linear protein. The core part is containing single polypeptide chain and side chain is made up of oligosaccharides. Two types of mucins are generally observed, membrane bound mucin and secretory mucin. Some characteristic of mucin includes like linear in its structure, it is flexible by nature and random coil-like structure it is. It is negatively charged due to the presence of sialic acid or sulfate residue in its structure, and it is cross-linked by disulfide bond. Disulfide form or a bond includes sulfur bonding, and it is attached with the donors form, and it is heavily hydrated. Mucous membrane itself has moist surface and lining the walls of various body cavities. Mucous membrane consists of a connective tissue layer above which an epithelial layer is available, which is mainly of single layer in case of if it is a stomach, small or large intestine or bronchium, while in case of if it is of esophagus, vagina or cornea, it will be of multilayered or stratified membrane. The thickness of this mucous membrane varies from 50 to 450 in case of stomach and lesser to the 1 micrometer in the oral cavity. This is the structure of mucous membrane in which outer part is made up of epithelium that represents the barrier to the penetration and basement membrane, the lamina propria and submucosa, which contains nerve and blood vessels. Epithelium may contain some keratinized or non-keratinized cell, and the drug delivery through the mucosa depends on mucin flow rate, pH of the environment, enzyme presence that are in the particular area, and permeability factors. So permeability again dependent upon the mucosal thickness, different epithelial cell composition and vascularization. So this brief introduction about mucous membrane, we will switch over to the different theories of adhesion. This theory of adhesion are specially assumed or derived for mucoadhesion purpose. Different scientists goes through different research work and their hypotheses gave generation of these different theories. These are the list of theories one by one. We will discuss it. First is the diffusion theory. Diffusion theory states that interpenetration and entanglement of polymer chain are responsible for this bioadhesion. Here the entanglement take place or penetration take place of the polymer into mucous network, and it depends upon the concentration gradient as well as the diffusion coefficient of the process. Here the penetration depth should be in the range of zero point 0.2 to zero point 0.5 millimeter for better mucoadhesion strength. Another theory is known as wetting theory, which is generally applied to the liquid system when the liquid is spreaded over the mucous membrane and it has to be attached and mucoadhesion take place. This theory is the base one. It is based on the surface as well as interfacial energies. The spreadability as a prerequisite for the adhesion because here liquid is dealing for the mucoadhesion. The spreadability coefficient SAB is calculated by gamma B minus gamma A minus gamma AB, where gamma A is
Madam,
hello, yes,
madam, are you changing the slides of the presentation?
yes, ma'am.
It is not showing on the screen.
Okay, again I am sharing.
Okay.
Now, is it wetting theory?
Yes, ma'am. Can you please try to change the slide so that we know it is working or not?
Okay, now diffusion theory.
No, ma'am. It is not showing here on the screen.
Okay, so
Uh, slide change not होती.
excuse me.
Yes, ma'am.
Actually, I am changing, but it is not showing in your side.
Yes.
Whatever I am uploading, that remains as it is. such type of problem you may be facing. Is it fracture theory?
Yes, it is fracture theory. Yes.
Then it is wetting theory.
No, if you are changing, then it is not showing on our side.
Now electronic theory.
No, madam. Uh, Ma'am, you can continue. We are doing the.
Okay, I will ask you for changing the slide if it is possible at your side.
Yes, yes, ma'am. You can continue, please, ma'am.
Okay, I will repeat from diffusion theory. Okay.
Okay.
So is it okay with diffusion theory at your side?
It is showing the last screen of fracture theory you showed us.
Okay, then I should repeat it from.
Let me remove and again add. It is still fracture theory, ma'am.
Because I am using application window and I am changing it, but something is wrong. Now,
Electronic theory.
okay, then now,
No, it is not changing. Please give me 1 minute, ma'am.
sure.
Hello, ma'am.
Yeah,
Ma'am, can you take out of the full screen and then try? Yes,
okay.
now after
Again, I
taking
should.
out, try without doing the slideshow.
Okay, then it's okay.
Okay.
Now, wetting theory is existing.
Yes, yes, ma'am.
Then fracture theory now.
It is not showing to us.
I'm selecting application window. Instead of it, should I select your entire screen or what?
Uh, you can try entire screen for this.
Let me try. But there is not possibility. Share button is not active for sharing at entire screen. And Chrome tab is also not usable. So only one option, that is the application window.
પણ મેડમને નથી શેર કરી હતી.
Now I have shared from application window.
Okay, it is showing now, but it is like if you change the slide, it is not showing. It is that way. You can select one and then go proceed with your.
So it means every time I have to withdraw, stop sharing, and then again next slide.
Uh, just a minute.
Now what is visible? Please tell me.
Madam, we have received the presentation and we would like to work from here and change the slide if that's
Better it will
feasible
be.
to you.
Better it will be.
Okay, okay. Please give me 2 minutes.
Sure. I have sent it to Chintan sir itself.
Yes, 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 ma'am. Ma'am, can you see the screen?
Yes.
Yes, then this is your presentation.
Yes. Sure.
Is it showing?
First slide is visible to me.
Okay, so I'm changing the slide. Is it showing?
Yes. Sure.
Okay, so
You
kindly
just
tell me in which slide you have reached.
theories of addition. Slide number 8.
Slide
Yes.
number 8.
Yes. Good.
Okay.
Good. Same.
So please continue. I'm muting my mic.
Okay. Thank you. So as I told, various scientists gave different theories based on their assumption and hypothesis. These are the list of theories like diffusion theory, wetting theory, fracture theory, electronic theory, mechanical theory, and adsorption theory. Let us discuss one by one in detail with first one, that is diffusion theory. Next please. Diffusion theory states that interpenetration and entanglement of polymer chain are responsible for bioadhesion. Here the penetration of polymer chain into the mucus network that depends on concentration gradient of the polymer and diffusion coefficient of the process. The penetration depth should be in the range of zero point 0.2 to zero point 0.5 millimeter for better mucoadhesion strength. Next please. Wetting theory. This theory is specially applied to the liquid system as it is to be spreaded on the mucous membrane, and at that particular place mucoadhesion is taking place. It is based on the surface and interfacial energies, and spreadability is a prerequisite for the adhesion, means liquid should spread on the mucous membrane so the adhesion can take place. It can be calculated as SAB is equal to gamma B minus gamma A minus gamma AB, where gamma A and B, that is the surface energy of the liquid A on and solid B, which are going to be involved in mucoadhesion process, and gamma AB, that is the interfacial energy between solid and liquid. Here SAB, that is the spreadability coefficient, should be positive for liquid to spread spontaneously over the solid. If this value is positive, it is beneficial for us for getting good mucoadhesion. Next, please. Fracture theory analyzes the force that is required to separate two surfaces after adhesion is established. Once a polymer or a dosage form is applied to the mucous membrane and mucoadhesion take place, then we have to apply the force to get separated, and that force required is measured by this particular theory. It does not take into account the interpenetration or diffusion of polymer chain into the mucous membrane for the calculation. Consequently, it is appropriate for use in the calculation for rigid or semi-solid bioadhesive material, in which polymer chain do not penetrate into the mucus layer. Here the figure indicates that fracture is observed in hydrated layer device. Same way fracture is observed at the interface and fracture in the mucus layer. So the dosage form will become separated from the mucous membrane. Next please. Another theory is electronic theory. This theory is based on a premise that both mucoadhesive and biological material possessing opposite electrical charge, and both materials come in contact with each other and they transfer the electron, leading to the building of a double electronic layer at the interface. Due to the opposite charge, they will be attracted and adhesion takes place, where the attractive forces within this electronic double layer determines the strength of mucoadhesion. Next, please. Adsorption theory, which describes the attachment on the basis of hydrogen bonding or van der Waals force. Adsorption itself indicates that it will be a surface phenomena where mucoadhesion takes place. When mucus is interacting with the solid dosage form, the molecules of liquid are getting adsorbed on the solid surface, and this is exothermic process where energy will be evolved and adsorption takes place spontaneously where delta G AD will be negative value. Next please. Another is the important phenomena called mechanical theory, which assumes that adhesion arises from an interlocking of liquid adhesive into the irregularity of a rough surface, which is a mucous membrane where adhesion is taking place. More the roughness, more will be the interfacial area where the interlocking will take place. So rougher surface will give better mucoadhesion properties, which is available to interaction and thereby aiding the dissipating energy, and it is most important phenomena of the process to get interlocked. Next please. These are the different steps of mucoadhesion. Mucoadhesion is not a single step, but it is a process completing in three individual steps, from which first one is the wetting and swelling of the dosage form or the polymer that is involved in the formulation. The wetting and swelling step occurs when polymer spread over the surface of mucosal membrane. Here the spreading occurs, and whatever liquid that is present on the mucus that will be passed on to the dosage form and wetting will occur in order to develop an intimate contact with the substrate. It is placed a bioadhesive formulation with oral cavity or vaginal cavity, and wetting will be observed. The swelling of polymer is observed due to the affinity for water by the polymer. Here, whatever polymers we are taking, they are hydrophilic in nature and they are having property to swell when they are coming in contact with the water. So, first step is wetting and swelling. Step 2, next please. Step 2 is interdiffusion and interpenetration. After getting swelled and becomes somewhat soft, interdiffusion and interpenetration takes place between the chain of mucoadhesive polymer and mucus gel network, creating a great area of contact. Here in figure, if you are seeing that bioadhesive polymer is coming in contact with the mucous membrane, they are just coming nearer and nearer, and the strength of this bond that are creating that depends upon degree of penetration of two of the polymer groups and if it is of same chemical structure, and one polymer if it is soluble in another one, that will give better bio or mucoadhesion strength. Next, please. Step 3, that is entanglement. Here the formation of chemical bonding takes place. Entanglement and formation of weak chemical bonding as well as secondary bonds between the polymer chain and mucin molecules are observed. This type of bonding formed between the chain of the polymer includes primary bond such as covalent bond and weaker secondary interaction bond forming, which are van der Waals forces interaction or hydrogen bonding. Figure indicates that there is an interpenetration between the polymer and mucin, which is transferred to entanglement where the chemical bonding like hydrogen bonding or van der Waals forces are occurring, and that's why it is called entanglement. Next, please. The next point to be discussed, that is the advantages of mucosal drug delivery system. First one is the prolonged residence time. Whatever dosage form we are preparing, it has a specific mucoadhesion strength, and for that benefit it is sticking to the area where it is applied, and that's why it is residing there for longer period of time. A localization of drug is observed at the target site where we have applied. At that particular site it will be localized, and termination of therapy is easy as it is applied to skin or buccal mucosa or ocular or nasal mucosa. It can be easily removed if any termination is required due to generation of some side effects. Systematic delivery of drug with bypassing the first pass metabolism is observed. If a drug is showing liver metabolism, it is possible by mucosal drug delivery to bypass the liver and prevent our drugs from first pass metabolism, and that's why it is offering a greater viability. A significant reduction in the dose can be achieved by reducing the dose-related side effect. As the dose is reduced due to the higher residence time, dose-related side effects will also be reduced. Unstable drugs like some of the drugs having acid liability or alkaline liability from the environment, we can switch over it to the buccal, sublingual or vaginal drug delivery. It is not compulsory to give through the oral route. The presence of saliva ensures relatively large amount of water for the drug dissolution. If it is a buccal drug delivery, saliva will be there in the mouth cavity, and in that saliva itself, water will be present and it will be providing some environment where drug get easily dissolved. Next, please. Some other advantages include systemic absorption is rapid, as it is absorbed through buccal mucosa or vaginal mucosa. Vasculature or blood vessel availability will be good one with that particular route, and that's why it will easily reach to the systemic circulation with faster effect. Alternative route for various hormones, narcotic analgesics, steroidal drugs, some enzymes or cardiovascular agents, etc. This route is very much helpful. The buccal mucosa is highly perfused with blood vessel and offers a greater permeability as compared to skin as a mucosal surface, so it can be preferable. Maximum utilization of drug is possible with the case of mucosal drug delivery, and that's why it is enabling reducing in a total amount of drug administered, and reduction in the plasma fluctuation is also observed as compared to conventional dosage form, because in conventional dosage form the dosing frequency is somewhat high as compared to this mucosal drug delivery, and that's why peak value fluctuation is reduced in this particular case. Next, please. Some of the disadvantage or limitation that is involved with the mucosal drug delivery system is like a drug which is irritating to the oral mucosa or having bitter taste or unpleasant odor cannot be administered through this particular route, and the drugs which are unstable at the target site pH. Say for example, suppose a drug is not stable at 7 pH or 6.8 pH, then it cannot be given through this particular route. So unstable at the target site pH cannot be administered through mucosal drug delivery. Only drugs with small dose requirement can be administered except GI tract. In GI tract, if it is given with bigger dose, it is okay. But if it is with the buccal mucosa or sublingual route, then smaller dose containing drug should be preferable. Another is only those drugs which are absorbed by passive diffusion can be administered by this particular route because here through mucosal membrane it is crossing by passive diffusion process. Eating and drinking may become restricted or patient has some uncomfortability in the case if it is a buccal mucosa. These are some limitation or disadvantage. Next please. Some other includes swallowing of the formulation. Patients who are not aware about sticking it in the buccal mucosa or keeping it in below the tongue, all that route are not known by the farmer patients, and that's why they may swallow. And this swallowing problem should be avoided by giving some instruction to the patient so it can be prevented. Overhydration may lead to the formations slippery surface. Sometimes it may happen that we are sticking the dosage form to the mucous membrane, but mucous membrane is releasing mucosa frequently, and due to that slippery surface is generated and overhydration may be giving a poor residence time, and with this slippery surface our dosage form will not stick properly. Structural integrity of the formulation may get disturbed or disrupted by swelling and hydration of the bioadhesive polymer. Next please. Where mucosal drug delivery system is possible. So these are the organs or place where mucosal drug delivery can be possible, like buccal mucosa, in the stomach, gastric attachment or adhesion, intestinal adhesion that is also possible, where the polymer should identify the pH or the after some 2 hours should get attached to the mucosa, etc. will be the target, and accordingly we should select the polymer. Then nasal mucosa, bronchial mucosa, vaginal mucosa, ocular and rectal where the mucosal drug delivery system is possible. Next please. Now, the transmucosal permeability. First and foremost requirement is the permeability. If drug is permeable to the transmucosal membrane, it is able to cross that membrane, then and then it will reach to the systemic circulation. Say for example, if ocular route, nasal route, or pulmonary route, rectal route, vaginal route, all these are the route from which they have respective mucosal system, that it has to cross that transmucosal membrane and enter inside through cross that membrane by penetration mechanism and entering into the general circulation. Same way, if it is given through oral route, gastrointestinal biotransformation may take place. Oral route may have this type of problem. So here we can have bypass of that route, and directly it is entering inside the just general circulation, or you can say systemic circulation, and it will reach to the target tissue through distribution by the blood media, and when it reaches to the target tissue it will show the pharmacodynamic response and a pharmacological action which is desired when it is observed. So that is the transmucosal permeability, which is prerequisite for good pharmacological response. Next, please. Some of the mucoadhesive polymers to be studied. So first we can say if polymers are very much important, that is because it has to adhere to the mucin epithelial surface and can be divided in three broad categories. First category is the polymer that becomes sticky when it is placed in with water and all that mucoadhesion to stickiness. Stickiness is achieved. Second category, that is the polymers that adhere through non-specific, non-covalent interactions. Those are primarily electrostatic or hydrogen bonding or hydrophobic bonding. Here, third category involves binding to a specific receptor. Specific receptors are only have ability to bind with the polymer and that is specifically designed for site-specific drug delivery. And these three categories of polymer are taking part in the mucoadhesion. Next, please. So before selecting the category, we should finalize the ideal category or ideal characteristic of the mucoadhesive polymer which we should be selecting for the generation of formulation. So first characteristic is polymer and its degraded product should be non-toxic and non-absorbable from the GLT. It should not provide any toxic effect after its absorption. It should be non-irritant to the mucous membrane. So many membranes are coming in exposure with this type of formulation. So any of the mucous membrane which is coming in direct contact with the formulation should not be irritated. Should preferably form strong non-covalent bonding with the mucin. So good bioadhesion is to be achieved. Should adhere quickly to moist the tissue. The strength that is to be achieved should be acquired very fast and it should be site specific where it is to be attached. Should not offer any hindrance or a problem to the drug release. If they are sticky in nature, it should not create any type of problem during drug release. Otherwise, it may retard the release of the drug also. Must not decompose on storage throughout its shelf life of the formulation. Whatever polymer we are selecting, that should be stable and it should not decompose whatever time period that is expected for the formulation. Should have an optimum degree of cross-linking, density, pH and hydration. All these properties should be optimum one according to our need. That is the aim. And finally, but very much important, that is the it should be economic because any industry that is dealing with the preparation cost and patient who is paying that particular money for the achieving their formulation, they all are expecting the dosage form should be cheaper one. So whatever polymers we are selecting should be economic. Next, please. So different polymers example I have taken. We can classify them into the cationic, anionic, non-ionic, ion exchange resin and some miscellaneous dosage form. Cationic polymer includes like chitosan, anionic includes polyacrylic acid derivatives, carboxyl, sodium alginate. Non-ionic where there is no chance for ionization like HPMC derivatives, HPC, HEC, cellulosic derivatives oils are coming under this category, polyoxyethylene, etc. Then ion exchange resin like polysteramine and miscellaneous include sucralfate, gliadin, etc. Next, please. So with the basic knowledge about the polymer and its characteristic, now we are switching towards the formulation consideration. These are the list of formulation. These are specifically for buccal drug delivery system like buccal tablets, buccal patches, gel, ointment, spray, in situ gel, powder, microparticles, microspheres, nanoparticles, etc. So these are the some of the doses form. Let us discuss one by one in detail. Next, please. So let us start with the buccal tablet. Tablets are small, flat, round or oval-shaped dosage forms specifically designed for buccal route of administration. The transmucosal tablets that are allowed for drinking and speaking without major discomfort. That is the basic requirement for generation of buccal tablet. They often become soft and adhere to the mucosa and then it will be retained in the position until the dissolution takes place or release of the drug completely. Monolithic or bilayer tablets are generally utilized. They are designed for buccal drug delivery. For this preparation, generally cup-in-pour method is selected, which is very famous and easy to manufacture at laboratory scale as well as in the industrial scale. So let us have the discussion about the preparation. Next, please. So cup in pour method includes generation of a dye cavity in which upper punch and lower punch are set in such a way with 3 or 4 diameter. So 4 tablet is prepared, which is presented here with orange color. This prepared 4 tablet is again placed in a bigger dye cavity which is having 6 or 7 diameter, and three side coating of polymer is done, and that's why one side is only open for releasing of the drug. Three of the side which is represented by green color, which is of baking layer, which is impermeable. So drug will release on a single side and it will be attached to the buccal mucosa. So this cupping core method is easy for preparation and give perfectness in the release mechanism and attaching to the buccal mucosa. Next please. Another dosage form is buccal patch. These patches are described as laminates which comprises of impermeable backing layer of a drug that contain a reservoir layer. Here, the reservoir layer containing drug should be supported by three side by backing layer which is impermeable just like a tablet, but here the thickness of patch will be somewhat lesser. So release of the drug in controlled manner is possible from bioadhesive surface to the mucosal attachment, and in opposite side we have impermeable backing layer, so it will prevent the drug to release it on the opposite side. For this particular preparation, solvent casting method is widely utilized to prepare adhesive patches. So let's have a view by solvent casting method. Next please. So this is the representation of solvent casting method in which a solution is prepared which is homogeneous in nature containing drug, polymer and solvent. A solvent is selected in such a way where polymer and drug both are soluble in it, and it is casted in a petri dish which is again kept in oven below 60 degree to get evaporated. Solvent will be evaporated for some time, and then film formation will take place after complete evaporation of solvent. After film formation, it will be kept that is kept outside, and then it will be dried completely and film will be stripped off with desired size range like 1 centimeter by 1 centimeter diameter, or it is 1 centimeter square or 1 centimeter round shape, whatever the size and shape we need, we can cut it and we can prepare it. Next please. Another dosage form is gel and ointment. Semi-solid dosage form having the advantage of easy dispersion of the drug, and it can be easily applied to the buccal mucosa. The problem of poor retention of gel at the application site should be overcome by adding bioadhesive polymers inside the formulation. Once bioadhesive polymer has been added and mucus transaction is controlled, then gel or semi-solid ointment will retain in the buccal mucosa for longer period of time. Here, viscosity is very much important. Certain adhesive polymers, for example, sodium carboxymethyl cellulose, undergo a phase change from liquid to semi-solid when it is applied. This change enhances or improves the viscosity of the dosage form, and it results into the sustainability and controlled release of drug is possible. Next, please. Next, another dosage form is spray. An aerosol spray is one of the suitable alternative to the solid dosage form because this spray can deliver the drug into the salivary fluid or on the mucosal surface, and it is readily available for absorption. Once we are spraying the dosage form, solvent will be evaporated and a thin film will be generated immediately. As the spray delivers the dose in a fine particulate matter or in a droplet form, the lag time for the drug to get absorbed, that is reduced, and faster effect can be achieved. That is the most suitable advantage of this dosage form. Next please. In situ gel. I think you have heard this word in situ gel, in which we have to prepare a dosage form which is liquid in nature, so probably solution will be there. And when the solution is incorporated into the body cavity, it will be converted to gel transformation is occurred at 37 degrees centigrade or sometime in the pH range of particular cavity like lacrimal fluid, buccal mucosa, etc. So the timely gelation and retention of the in situ gel formulation could be the fundamental in improving the efficacy of the drug. It is easy to administer and then retention is observed. The phase changes polymers are used for, for the preparation of thermoreversible gel. Here, when it is incorporated into the aqueous solution form, this polymer exhibits salt to gel transition in response to the body temperature. Again, the pH or specific ions, if it is present, then it will be the mechanism for converting salt to gel. And it allows advantage of easy topical application also. Next, please. Another is powders and microparticles. Powders or microparticles are generally prepared by simple mixing of drug substance with excipients. It can be achieved by spray drying, freeze drying, or direct production of drug that is loaded into the nano or microparticles. Nanoparticles or microparticles are prepared and then drug is to be loaded on the surface of this particle. Next, please. Next, then, how to prepare this microsphere or the nanoparticle? They have advantage of being applied in a liquid form, just like eye drops. The nanoparticles can be dispensed in form of nanosuspension, and then it is easy to deliver at the particular site. And this suspension contains mucoadhesive polymer, so at the site of sticking or a site of action, it will stick to the mucous membrane and adhesion is possible. They avoid the discomfort that combined with the application of viscous or sticky preparation like ointment. They have some advantages like targeting of a drug to the site of action or leading to a decrease in the dose which is required because maximum drug will be utilized and that's why reduction in the side effects can also be achieved. Next please. Now, all these are the formulations that we have discussed, and only the formulation aspect is not enough. We have to check with some evaluation parameter regarding the mucoadhesive strength. So, if we have prepared a tablet, we have to check the weight variation, friability, some other parameters also. But side by side, as it is a mucosal drug delivery, it should be checked for mucoadhesive strength. Same way, other parameters to be checked. So here, due to time constraint, I am only discussing the parameters which are specifically for mucosal drug delivery. Rest of the parameters will be common, same as the conventional doses form. So first parameter to be studied, that is in vitro test or ex vivo method, that is based on measurement of tensile strength or mucoadhesive strength. Here, in this method, force required to break the adhesive bond between the model membrane and the test polymer is to be measured. Here what happens, two jaws that are prepared from a flat glasses are pulled free from the upper glass and here both the glass are adhered to the tissue which is a biological membrane to which a mucoadhesive dosage form is applied and then they are pulled in an opposite direction. So, how much force is required that is to be calculated and recorded as the mucoadhesive strength. Next, please. In vitro drug release study. Muco retention ability of the studies include study related to the polymer and how they are releasing the drug. A 1 centimeter by 1 centimeter piece of porcine buccal mucosa is tied on a glass slide. Glass slide which is used in our laboratory generally 3 inch by 1 inch in diameter so size range and it is tied on slide through thread and a tablet was stuck on the wet rinsed tissue by applying a light force with a fingertip for 30 seconds. We have to press that particular dosage form on this mucous membrane and that mucous membrane is tied with a slide and the slide is kept on one of the grooves of USP tablet disintegrating test apparatus. And from that, we have to operate this disintegration test apparatus for a desired period of time and predetermined time interval. We have to collect the sample from the media, and we have to analyze it for the presence of a drug inside the sample. So that will give us about how much amount of drug that is going to be released, and it can be analyzed with suitable analytical method. Next, please. In vivo method includes use of radioisotopes. For example, we are using barium sulfate that is encapsulated in the bioadhesive polymer for determining the bioadhesive strength and presence of the drug inside the GI tract. So simple procedure that use radio-opaque marker that can be given to the patient, and then we can mark or we can check the presence of this marker in the different areas and their concentration, and depending upon that we can check the in vivo availability of the drug as well as polymer. Next, please. Use of gamma scintigraphy, information of oral dosage forms across the different region of the GI tract. Here, it can be achieved by gamma scintigraphy. So, which type of information this gamma scintigraphy gives us? First one, the time, and second one, the site of the disintegration of the dosage form. At which particular site it was converted to the particulate form, the site where the drug was absorbed, and the effect of food, presence of some disease in the patient, and size of the dosage form. All these are somewhat maybe creating some problem in the dosage form. So it can be studied by gamma scintigraphy. So in vivo performance of the dosage form in combined way, we can say, can be done by this particular method. Next, please. So this is all related to formulation and evaluation. We are reaching to the final stage of the presentation, which is conclusion. What type of conclusion we can derive from this particular discussion? First thing, that is the transmucosal route can address the limitation by traditional oral route and parenteral route. Some of the limitations are there with the conventional dosage form that are given by oral or parenteral route, and it can be overcome by this particular selected drug delivery system. The close contact of the formulation with biological membrane can improve the transportation possibilities of a drug, can easily transfer the drug through the mucosal membrane. It can penetrate and reach to the systemic circulation very fast, as well as viability can also be improved. Larger molecule can be given with permeation enhancer, and some transmucosal membrane can further be studied in detail for the safe delivery of protein or peptide-like drug molecules using different formulation approaches. Here, this is the research area where protein and peptide drug molecules, bigger in size, are a challenge for us to the formulate mucosal drug delivery for this category of the dosage form. Next, please. So thank you everyone for patience listening, and I'm very much thankful again to the organizing committee of the Subhas Technical Campus who gave me the opportunity to share my views on this particular topic with all of you. Thank you all of you once again.
Thank you so much, ma'am, for your informative session. We have few questions that are asked by the contestant participants. So here is the first question.
Yes,
Which mechanism is used for mucoadhesion by using mucoadhesive polymer? Any specific mechanism available?
I didn't get your question. Can you repeat, please?
Which mechanism is used
Yes.
for mucoadhesion by using mucoadhesive polymer?
Okay,
Any specific?
mucoadhesive polymer are just get wet first, then get swelled, and due to swelling they become softened, and due to the structural activity of that polymer, they will bind with the mucosal membrane, and that membrane will get entanglement. Means penetration is very much important for getting better mucosal mucoadhesive strength. So that is the basic mechanism. Swelling, then comes consolidation, and then interpenetration.
Thank you, ma'am. The next question is mucoadhesive drug delivery increases permeability of dosage form. How is it possible?
We have to add some permeation enhancer if our dosage form have any problem with the permeability. So that is the limitation where addition of permeation enhancer is required for specifically BCS class 3 drug and 4 drug where permeability is an issue.
Okay. And the next question is it mucosal drug delivery system more expensive as compared to other drug delivery system?
I don't think that much expense is there because the polymer which are using that is not that much costly as compared to nanoparticle generation and all. And advantage is whatever they are providing to us that is very much important. So the cost which is increasing is not much more.
Okay. Thank you, ma'am. And one last question for today. What is the future of MDDS?
In future, if I say generation of a dosage form or formulation of protein delivery or peptide delivery, where we have to work, where research work is going on, and this type of bigger molecules, if they are easily given by this easy drug delivery, that is requiring some research area. So we have to work on that, and there are some chances for the research.
Okay. Uh, Thank you so much, ma'am, for such an informative session on mucosal drug delivery system. I am very thankful to all the participants for attending this webinar, and I am also thankful to our management and our director, Dr. Deepak Patel, sir, for their continuous support to organize this webinar. Once again, thanks to all of you. Have a good day to all the participants, and thank you once again, ma'am. Thank you so much.